I will introduce our next speaker is Dr. Michael Sacher from Concordia University in Canada. In studying TANGO2, his lab has established several model systems, including fruit flies and human fibroblasts, to aid in their work. These studies have led to the discovery that vitamin B5 alleviates cellular defects associated with non-functional TANGO2. And Dr. Sacher currently serves as a member of the TANGO2 Research Foundation's Board of Directors and the Scientific Advisory Board. Please welcome Dr. Michael Sacher.
All right, well, thank you very much. Pleasure to be back here. I was here 2 years ago, so I'm seeing some familiar faces, which is good. So I'm seeing some faces that I usually only see in 2 dimensions on Zoom, so it's good to see a 3-dimensional look and some new faces as well. It's great to see new people here as well. I realize I have the unenviable task of standing in front of a room full of people who are waiting to eat lunch. So I'm going to try my best to keep you focused on my presentation and a little less on your growling stomachs. So I'm going to just to let you know, I've received some funding generously from the TANGO2 Research Foundation. And in terms of disclosures, I'm a member of the board of the foundation and the Scientific Advisory Board. But the biggest disclosure that I have to reveal is I'm a diehard Boston Celtics fan. So if I seem a little giddy from the championship last week, then it probably is the case. And I guess the same holds true for Mike, Ryan, and everybody else from that area. And we won't talk to the people from Texas. Okay, so I'm going to give a very short background because there's been great backgrounds already today, and I didn't think I would need to give too much background. Then I'll get into some results. I'll show some of the, there's a pointer on here somewhere. Yeah, I'll show some of the vitamin B5 work that we did on flies and how we've extended that now into human cells in terms of looking at lipids in those human cells. And along the way, you'll see some hypotheses that we have and how we go about testing them or how we plan on testing some of them. Okay, so just thinking back to your high school science class, there's a few compartments that I'm going to talk about. The endoplasmic reticulum very briefly. Proteins that get inserted into the endoplasmic reticulum are often transported to a compartment called the Golgi. So I'm only going to briefly talk about that pathway. Mitochondria, I'm sure everyone is familiar with, the powerhouse of the cells where most of the energy is made. And I'm going to talk about another compartment today a little bit more called the lipid droplet. Okay, so in terms of background, this is pretty much all I want to give because you've heard a lot already. It was discovered nearly 20 years ago at this point in a screen that was looking for proteins that were involved in transport to or organization of the Golgi, and then hence the name TANGO. And so about 20 different genes were discovered. One of which is TANGO2. The point here is that if we know the function of any one of those other 19 genes, it doesn't necessarily mean we know anything about TANGO2. Okay, they just have the same name because they were generated from the same screen. So can we tell anything about TANGO2's function from its structure? So this is the predicted structure of TANGO2. Proteins are made up of a string of amino acids, but that string is then organized into 3 dimensions. And so this is what TANGO2 would look like is predicted to look like in 3 dimensions. And then what we do is we take that structure and we ask, do any other proteins have a similar type of 3-dimensional structure? And if they do, what is the function of those other proteins? And then maybe we can start coming to a function for TANGO2. And it turns out that there's a number of different enzymes that have similar portions of them that resemble TANGO2. For example, phospholipase enzyme, which is an enzyme that cuts fatty acids off of lipids. An N-terminal nucleophile, or an NTN hydrolase domain, is a family of proteins that shares some structure with TANGO2. But it's not really telling us exactly what TANGO2 would do. However, We suspect then that TANGO2 must have some sort of enzymatic activity. All right, so just sort of file that back because I'm going to return to that towards the end. I don't think I need to show clinical features of children with TANGO2, but I'm showing this because I'm going to show you some work that we did on flies, and the flies share a lot of these features. So there's metabolic features, neurologic, developmental ones, and you'll see that for many of them, we've been able to replicate that in the flies, and in all cases, we've been able to rescue those with just treating with vitamin B5. Okay, so we started off with a hypothesis that TANGO2 is regulating lipid metabolism, and that could help explain why starvation or removing sugar from the diet could induce a metabolic crisis. So how are we going to test that hypothesis? So we were going to use vitamin B5, as Sarah already mentioned in her talk. There's only one function of vitamin B5 that's known in cells. It goes through this 5-step pathway to make this molecule called coenzyme A, which is required for a lot of lipid metabolism. Okay, so we decided to take a fruit fly model that has a mutation in TANGO2 and to also look at some human fibroblasts taken from a child suffering from TDD, TANGO2 deficiency disease. So again, this is just the 5-step process of taking vitamin B5, also known as pantothenate, to this molecule called coenzyme A, and again, it's the only thing known for vitamin B5 in a cell. So the first thing we did was we starved the flies. We put them in a plastic vial, no food, and we just looked at their survival rate. You can see they're dying off. That's in blue is the TANGO2 flies. All the flies are dying off. The controls are shown in black, but you can see the TANGO2 flies are more susceptible to dying off much faster, I should say, than the control flies are. However, when we supplement those flies with vitamin B5 before doing the starvation, that's shown in green, we now shift that curve to the right, so those flies are surviving a little bit better. It was one of our first clues that vitamin B5 might actually be doing something important with TANGO2. We looked through the developmental cycle of these flies. They start off as a little egg, they turn into a larva, then they develop into the adult fly that we all, I won't say know and love because we hate them when they're flying around our kitchens, but they develop into those flies. So this is at the larva stage, and we can watch these larvae crawl around a dish. And so this first black line here is just looking at the control larva, what distance do they move over a given amount of time, and compare that to the TANGO mutants, you can see that they move much less, these mutants. However, when we supplement those larvae with vitamin B5, but not with any of these other vitamins that we looked at, and there's more vitamins that we looked at as well, you can see that it's rescued to the level of what's seen in controls. So another benefit of vitamin B5. We went to the adult flies now, and in this geotaxis test, what we do is we put them in a plastic vial, smash the vial on a table, the flies all fall to the bottom, and then we let them crawl up the sides. And we mark off a little finish line, and we want to see how many of the flies can cross the finish line within a 10-second period, a very short period of time. Okay, so about 80% of the control flies can reach that finish line. The TANGO2 mutant flies, only about 15% of them do. But again, when supplemented with vitamin B5, but not with any other vitamins that we looked at, we can rescue that phenotype as well. We also looked at seizures. Flies, we can induce seizures in flies under certain conditions when certain genes are mutated by just simply raising the temperature. You can see control flies don't experience a seizure when we raise the temperature to 42 degrees Celsius. About 60% of the TANGO mutants do experience a seizure. Last time I showed a video of a fly having a seizure. I thought I would, if you want to see it, I still have it, but you know, could be a little disturbing, I guess, to watch a fly have a seizure. But well, though we saw a zebrafish have a seizure, didn't we? So maybe I should have kept it in. So about 60% of them have a seizure. It's very easy to recognize, but when they're treated with vitamin B5, the number of flies that have a seizure is greatly reduced, and the time that it takes to see that first fly have a seizure is increased to 90 seconds, whereas when the flies are not treated with vitamin B5, it takes about only 30 seconds for us to see that first seizure. One of my favorite experiments actually was this one, where we now started asking, do the flies, these adult flies that we did that geotaxis assay on, where they crawl up the sides of the vial, do they have to be treated with vitamin B5 for their entire life, which was really 10 to 12 days at that point, but do they have to be treated the entire time, or can we just give them a little shot of vitamin B5? And so here's the mutants that aren't treated. Again, about 15 to 20% reach the finish line within 10 seconds. And you can see that even as short as 1 day before the experiment, there's a significant increase in the number of flies reaching the finish line, and that continues to increase with each day of treatment. And so that was showing that at least for a fly that has a model of TANGO2 deficiency syndrome, it's not too late to start vitamin B5. And there's benefits quite readily. It's actually borne out, I think, in some recent papers that are appearing that and anecdotally it seems as well that just taking vitamin B5 or B complex, I should say, is showing benefit regardless of age, it seems. Okay, so this is that figure again, and I've checked off things that we've tested. I haven't shown you our learning assay or the ataxia that we had done, but all of these are seen in the flies. They're all rescued by vitamin B5. Some of them we can't really readily look at. I mean, speech in a fly, right? I mean, maybe after a few glasses of wine we can start hearing flies talk to us, but we're certainly not sober, at least be able to tell that flies are talking. Okay, so we then went into the fibroblast system, which cells taken from an individual with TANGO2 deficiency disease. Now this is a complicated looking slide, but a graph. What we're looking at is the transport of material from the endoplasmic reticulum to the Golgi. We look at that over time, how it builds up in the Golgi over time. The solid lines are all control fibroblasts that have been treated with various vitamins. The dotted lines are all those TANGO2 deficient cells treated with various vitamins. And what I want to point out is how all the solid lines cluster together at a faster rate of material arriving in the Golgi versus these dotted lines from the TANGO2 cells, except for one dotted line, which you can see, that red one, which is clustering with the controls, and that is the TANGO2 deficient cells treated with vitamin B5. Okay, so it seems to be doing something in cells as well. Okay, so now our hypothesis is, well, since vitamin B5, this is a precursor of coenzyme A, and coenzyme A is sometimes referred to in the literature as a lipid activator. Lipids are bound to coenzyme A before they do something else. So maybe that's going to alter the lipidome, which is just the collection of all of the lipids that are found in a cell. So we're going to test that by performing a lipidomic analysis on TANGO2 deficient cells compared to cells that have the TANGO2 gene. But first we wanted to look at whether we can support the idea that TANGO2 might have something to do with lipid metabolism by asking, do the levels of the TANGO2 protein itself change as the cells shift from not utilizing lipids to utilizing lipids? So this is a Western blot, which is a method where we can just look at the TANGO2 protein. So this represents the TANGO2 protein at different times after we starve the cells. So we've taken away glucose, and now these cells are undergoing some sort of metabolic stress, should be utilizing lipids more readily, more frequently, and you can see that the TANGO2 levels are rising during that starvation. Doesn't mean that it's involved in lipid metabolism, but it's certainly consistent with the notion of it being required under some sort of metabolic stress. Okay, so this is, again, a little bit of a complicated looking slide, but these are different classes of lipids. And what we're looking at here is the ratio of those lipids in TANGO2 deficient cells compared to control. So for example, if we look at this one here, this is triacylglyceride. We found that it was 6-fold higher in the TANGO2 cells compared to the control cells. A number of other lipids were elevated as well in the TANGO2 deficient cells. This is under the normal conditions, normal fed, so they have glucose there. When we removed glucose and starved those cells, now you can see a lot more lipids are starting to accumulate in those TANGO2 deficient cells. So it is suggesting that there is some sort of lipid imbalance that's taking place, particularly during starvation in the TANGO2 deficient cells. So what happens when we look at this data after treatment with vitamin B5? You can see it's pretty much all rescued. The triglycerides are still high, but if I go back a slide here, it was 6-fold or 8 or 9-fold under starvation. It's much reduced after vitamin B5 treatment. I'm going to skip that slide for the purposes of time. Now, a number of lipidomic studies have been done over the last year or so. Vandana talked about the zebrafish study. We've done human cells. We've done fruit flies. We haven't published it just yet. The Malhotra lab, Agu, was doing the HepG2 cell line knockdown. And so what we were thinking is if we looked at all of these studies, maybe it's going to be like a Venn diagram where there's going to be some intersection here and there's going to be some specific lipids that are dysregulated, higher or lower in all of these model systems. Yeah, there's going to be a whole bunch of other things that are going on. Maybe those are not quite of interest, but maybe if we can find those lipids, handful of lipids that are in common to all of these, that would be something. Unfortunately, we didn't really see any lipid signature. If you think about the triglycerides, we see elevation of triglycerides. Vandana's fish have lower triglycerides. So we've decided to pursue the triglyceride angle just a little bit anyways. And so triglycerides are stored in a compartment referred to as the lipid droplet. And so what we wanted to know is, does TANGO2 associate with the lipid droplet, particularly under conditions where the lipids are going to have to be metabolized from those lipid droplets? So this is just a measure of the overlap between TANGO2 and lipid droplets, whether TANGO2 is associating with the lipid droplets. So this is under control conditions. We can force more lipid droplets into the cell by treating it with a fatty acid called oleic acid. So we treat them with oleic acid, we get more lipid droplets, and we see a slight increase in the amount of TANGO2 that's interacting. Not a very high amount, admittedly, but significantly increased. But now when we withdraw the oleic acid and start starving the cells, where now they're forced to get their energy from fats, and they're going to start metabolizing the lipid droplets, we see an increase in the amount of TANGO2 that associates with the lipid droplets. It peaks after about 1 hour and then starts to come back down. Okay, so it seems like a small pool of TANGO2 can associate with lipid droplets. We also measured some features of the lipid droplets, so you can see that in the TANGO2 deficient cells, there are many more lipid droplets seen than in the control cells. When treated with vitamin B5, we rescue that. We also saw that there was an increase in the volume of the lipid droplets. There's not just more of them, but they're bigger. And when we treat with vitamin B5, again, we rescue that, those lipid droplets. There's fewer of them, and they're back to more of a normal size. So if we just sum up what's happening with vitamin B5 work, it's rescuing all the phenotypes that we've seen in the fruit fly model. It's rescuing the ER to Golgi trafficking defect that we saw in the human fibroblasts. It's reversing that lipid imbalance that we saw in the fibroblasts, the human cells. And it's these changes that we're seeing in lipid droplets, lipid droplet number, lipid droplet size, is also being rescued by vitamin B5. And it's interesting to note, we heard just a little bit about 22q11 deletion, where one copy of TANGO2 is missing, and if a second copy has a pathogenic mutation, that individual could be suffering from 2 diseases. And perhaps vitamin B5 might be of use in those specific types of individuals, or individuals that have mutations in transporters of vitamin B5 itself or coenzyme A, where the phenotype is actually, the clinical phenotype has a lot of similarities to TANGO2. So that's something to consider for the future, but for the TANGO2 community, at least our work suggests that vitamin B5 is an important vitamin to have. So what does all that mean, right? Does anybody remember Double Rainbow Guy, or is that like just me having fun when I'm getting my slides together? I think it's just me having fun getting my slides. Remember that guy when he saw the double rainbow? And yeah, right across the sky. What does it mean? He kept, anyway, so I'm not nuts. So what does all that mean? Does that mean that TANGO2 has something to do with lipid droplets? It might have something to do with lipid droplets, but we think the story is a little bit more complicated than that. Now, you've seen this pathway before, and Sarah was showing this again in her talk as well. This is where vitamin B5 enters the cell and gets converted to coenzyme A in that 5-step pathway. And the levels of coenzyme A have to be maintained or balanced within a cell depending on physiological conditions. And so the cell has a way of degrading coenzyme A, and it can be degraded all the way back to vitamin B5, or it can be degraded part of the way back, and then it can be rescued or salvaged from some of these intermediates as well. Now, it doesn't seem that TANGO2 is going to function in this part of the pathway because, for reasons, again, that Sarah mentioned, vitamin B5 is working. And if vitamin B5 produces coenzyme A, there likely is no defect in this part of the whole process. In addition, there are individuals that do have mutations in some of these enzymes that synthesize coenzyme A, and they have signature type of phenotypes that are not seen in TANGO2 deficient disease. And so that leaves this degradation pathway or the rescue of these intermediates as a likely spot where TANGO2 may be functioning. Okay, so it seems that TANGO2 might be involved in the degradation or the salvage. Okay, so how are we going to test that? So we can measure the levels of these metabolites, all of those. I'll go back a slide again. Anything here that you're seeing here, we can just refer to as a metabolite. It's just something that's being produced during a cellular process here. So we can measure the levels of all those metabolites. We can measure the levels of coenzyme A in our fruit fly model, in our human cells, and if this meeting was a few weeks later, I would be more comfortable showing the data that we just received last week from the metabolites from the human cells and the coenzyme A from the human cells. So our coenzyme A results are very similar to the worms, where the levels of coenzyme A drop only during starvation, not in the non-starved cells. And a number of these metabolites are showing differences in the TANGO2 cells compared to the control cells, even prior to starvation as well. Okay, so that just to say that I think we're not barking up the wrong tree when we're talking about that whole pathway, that degradation and salvage pathway. So then we're doing, a lot of the reactions that you're seeing over there are enzymatic reactions. And so we're setting up enzyme assays to see if TANGO2 is doing any of these, involved in any of those steps that are in that degradation or salvage pathway. And another result that hopefully we'll get within the next few weeks, because it's been quite a long time coming, is this result of what's called a MIDAS assay, where we look for metabolites that bind to TANGO2. And we're hoping that what we see is going to complement what we're seeing in terms of the metabolite changes, and the two together will give us a better picture of where TANGO2 might perhaps function. Now, this question may have been gnawing at your minds for the entirety of my talk. I'd be surprised, actually, if it did. But it does gnaw at my mind because we've worked with yeast before. And the question is, can we use another model system? Let's throw another model system at everybody here, the yeast, the same yeast that you use for bread or for beer. Can that be used? Why would we want to use it? Well, number one, it's quite a simple system. There's only 6,000 genes there. and TANGO2 is well conserved. And so there is a TANGO2 homologue in yeast that really hasn't been looked at. And so the answer to that is yeah, for sure we can look at TANGO2 in yeast if we can tease out some, or if those yeast have a phenotype. We need to be able to see something different about those yeast so that we can now try and correct it. Okay, so what I'm showing here is we've taken 3 different yeast strains and knocked out TANGO2. The homologue is called YGR127W. We've knocked it out in 3 different strains. Absolutely no phenotype. What we're looking at here is just the growth of the cells. That's the typical phenotype that a yeast biologist would look for. Just do they grow poorly? Maybe if I change the temperature, if I change the growth conditions. But you can see in all these cases, the blue, the red, and the black, those are just our 3 different backgrounds with and without the TANGO2 gene. But now you're looking at these green ones, you go, well, there's something with a phenotype. Well, yeah, there is something with a phenotype there. That came from that Sun et al. Paper, which we've now seen 2 people talk about in terms of C. elegans and zebrafish not being able to reproduce some of those results. So we also got the yeast strain that they made from them, and sure enough, it has a severe phenotype. You can see it's growing pretty poorly. One experiment that every yeast biologist will do, when you see a phenotype and you think it's due to my favorite gene, I'm going to put my favorite gene back into those yeast cells and show that now they grow normally, right? So we did the experiment for them. For some reason, it wasn't done, and it didn't help at all. Would that suggest to us that whatever this growth phenotype is due to, it has nothing to do with the yeast TANGO2? All right, so if that's the case, then, well, we've got a bit of a problem here. We've got yeast cells where TANGO2 is knocked out, and how do you propose to work with that? Well, we've been playing around with conditions a lot, and I think, and maybe we can think about doing this meeting late July next time, because maybe in another few weeks we should know. I think there's not only 1, but maybe 2 conditions that we have where we can tease out a phenotype in these yeast the yeast TANGO2 knockouts. And if that's the case, I find that pretty exciting because we've humanized those yeast, meaning we've taken out the yeast TANGO2, we've put in the human TANGO2. And if that can allow those cells to now grow properly, that's a nice little system to study the human TANGO2 gene in. We've humanized it. But we can also justify studying the yeast TANGO2. If they're functioning the same way, then why not just study the yeast TANGO2 in the native yeast background? And we can use those yeast cells with that phenotype to do both genetic and chemical screens to find other genes that are involved with TANGO2. What pathways does TANGO2 function in? And pretty excitingly, I think we can use a small molecule library for small molecules that can functionally replace yeast TANGO2. Right now, if you think about that last sentence, a small molecule that functionally replaces TANGO2, we already have such a thing, right? It's called, has nobody been listening to me? Vitamin B5, right? Doesn't vitamin B5 seem to functionally replace TANGO2? When no TANGO2, we put vitamin B5 in, things seem to be going back to normal, okay? So you might ask, okay, so why do we need to look for something else? Well, that's a good question, and we can certainly discuss that afterwards. It requires a lot of time and money to do that. From a basic science standpoint, I think it's a great thing because if we can find the target of that small molecule, we can have also a better idea of what TANGO2 might be doing in those cells. But perhaps another small molecule will be beneficial instead of vitamin B5, in addition to vitamin B5. That's way down the road. Okay, so that's all I have. I'm just going to thank everyone who contributed to the work. Maza is over here. She did all of the lipid work. Alia is over here. She's doing the yeast work. Rosemary has taken over the fly work. That's Rosemary here in the yellow, and many of you remember Perea. She was here a couple years ago. She did all of that initial vitamin B5 work. as well. This is not your typical Wednesday afternoon in my lab where we just take out some wine and cheese, and it's actually right after Priya's convocation, and she was elected to give the valedictorian address at convocation as well. So we were celebrating all of that. So, and I haven't had time to talk about any of the work my collaborators are doing in establishing a knockout, TANGO2 knockout fly and a humanized fly as well. So anyways, I look forward to speaking with many of you later on. Thank you.